So we are going live on YouTube. Hello and welcome to the seventh talk of Science Month, a science conference organized for students by students. And today we have a really special guest. And I request Tanishta from first year to go ahead and introduce our guest.
A very warm welcome to all of you out there. This is the seventh talk of the Science Month organized by the Campus Radio team and the Science Club of ISA Kolkata. Today we have amongst us Mr. Dhiman Ray, who would enlighten us on looking at molecules through computational microscope. Mr. Dhiman Ray is currently a graduate student in the Department of Chemistry at University of California. He completed his integrated bachelor's and master's degree in chemistry at ISA Kolkata. His research interest is theoretical chemistry, particularly in the study of statistical mechanics and molecular dynamics simulations to study properties of biomolecules in condensed phase. He is also interested in developing new mathematical and computational methods which will help us get a deeper understanding of molecules and give better predicting power. Apart from research, he is also enthusiastic about food, traveling, and photography. Mr. Dhiman, we welcome you once again. I hope our audience will have a wonderful time. I would humbly request them to post their questions in the YouTube live chat. Thank you. Over to you, Mr. Dhiman.
Thank you very much for giving me this opportunity to talk to you about some of the interesting things which make me excited about computational chemistry. So I am Dhiman. I am from the Department of Chemistry of University of California, Irvine, USA. So in this talk, I will give a broad overview of the computational chemistry and its applications in different parts of chemistry and other scientific questions. So the title of the talk is Looking at Molecules Through the Computational Microscope. I will begin with a brief introduction about myself, and then I will give an idea about what is computational chemistry. Then I will move on to discuss about some of the recent success stories of computational chemistry in solving some real life and also scientific problems. Then I will touch upon some of the work that I performed as a part of my PhD, and then I will give a brief idea about some of the new frontiers and emerging areas of computational chemistry that will become, or most likely will become, a very big fields in future. So I completed my integrated BSMS from ISA Kolkata at the Department of Chemistry, and in my master's thesis I worked with Professor Roshni Tiwari, and during this time I got acquainted with the field of computational chemistry because I did my research on using quantum mechanical methods to predict new catalysts or design new catalysts based on materials. Then after finishing my BSMS, I moved to USA to do my PhD at University of California, Irvine, with Professor Iwan Andrecioi, with whom I'm currently working. During my work at ISA Kolkata, I was fortunate enough to do 2 summer internships. One of them is at Max Planck Institute for Coal Research in Germany with Dr. Matthias Hayden, and another one at University of Wisconsin-Madison in USA with Professor Chang Shui. In those 2 internships, I got acquainted with a different field of computational chemistry, which is the computational biophysics. This particular field deals with studying biomolecules in condensed phase and in solution phase and understand their behavior as it would happen inside a cell from a dynamical point of view. So I fell in love with this particular area of computational chemistry, and now my PhD is mostly concerned in that area. So what is computational chemistry? As you know, chemistry is mostly an experimental science, but computational chemists try to use theoretical models to understand the behavior of the molecules. So this process goes into multiple steps. First, one should write down the equations which will describe the electronic and also the nuclear properties of the molecule. And once you have the equations, then you need to put the equations in the form of a code or a computer program which will be able to solve this equation in a feasible manner, because in most cases these equations are so difficult that analytical solutions do not exist. Then from the code, it will going to predict the molecular properties, which includes the structure, some uh, charge distribution, and some other properties like dipole moments and stuff like that, and also the dynamics and the interaction of different molecules, which we can visualize in the form of molecular modeling softwares which are available for different computing systems. Now, looking at this, you might think that this is a process which a computer scientist should do. So, is this chemistry? Because the what the theoretical chemist or computational chemist is doing is basically concerning themselves with a lot of equations and things like that. So, to resolve that doubt, let's see that how that computational chemistry is helping in the progress of the science or the chemistry itself. So the most recently, the in 2013, this field has been recognized with the Nobel Prize with Professor Martin to given to Professor Martin Karplus, Michael Levitt, and Arieh Warshel. So in this Nobel Prize citation, it was mentioned that this computational chemistry is taking the experiments to the cyberspace. So here you see an experimental scientist or an experimental chemist who performs some synthesis or maybe find out the structure of some molecule, and then he passes on that information to a theoretical or a computational chemist. This person does theoretical analysis or computational calculations and predict new reactions or new molecules or new properties of the molecules which were not known before, and then he passes on that information, you see, to the experimental chemist, who again perform more experiments to verify those or to nullify some of those claims. And once these results come, they again goes to the computational chemist for further analysis. So in this way, as the Gunnar Karlström from Nobel Committee has correctly pointed out, together they climb the never-ending stairs of chemical knowledge. So the computational and the experimental part complement each other to get a much better and a complete understanding of how chemistry takes place in different situations. So where is computational chemistry used? And to be frank, if I want to give a one-word answer, then it's everywhere. Starting from modeling solid materials, which for gets can which are applied in solar cell, battery, and superconductor, etc. And in those cases, one would use computational techniques, for example, like density functional theory, and that sort of quantum mechanical models to study their behavior. There is also the system of organic and inorganic reaction mechanism predictions or catalyst design, which mostly depend on the the idea of the molecular orbitals and the electron densities and their movement and change throughout time. These are also done using quantum mechanical methods. On a slightly different area, uh, in the application to physical chemistry, where quantum dynamical techniques can be used to predict the spectra of small molecules or materials, and then these spectra can be compared with the actual experimental spectra for a better analysis and interpretation. These three portions are mostly from quantum mechanics, but one can use statistical mechanics or classical mechanics to understand systems of very large size, for example, a complexation of polymer and soft material systems, and also the biomolecules for the purpose of understanding their behavior inside cell and also for generating biomaterials and drugs. As I am particularly doing my research in the field of biomolecular simulations, this rest of the talk will be focusing mostly on this area. So the experiments that we mostly know, which are performed in the biophysics or biomolecular chemistry, they can predict mostly two sets of properties. One is the molecular structure and the other is the bulk properties. For example, if you see here, that if you do an X-ray diffraction experiment or a cryo-electron microscopy experiment, you can see the electron density that you can obtain from that, which is shown in the green color. Fitting the electron density, one can actually obtain the structural data of how the molecule looks like. But this is a very static picture, and the dynamics of the molecule is missing. We cannot see that how the reaction is happening or how it is moving and things like that. On the another extreme, the experiments also predict some bulk properties. For example, this is a uh, binding constant of a protein and a small molecule ligand, or maybe the rate constant of a folding and unfolding of a protein. So these are specific numbers which we can get from a experiment, and these are very low resolution picture because we just know the particular number and not the molecular level details which are giving rise to that particular value or the mechanism. To give an analogy with the football, which many of my American friends will call soccer, experiments is primarily like looking at the formation of the players before the beginning of the match. This is the static picture which we can get from the structure elucidation methods. Also, in the another spectrum, we can get the final result. We know that who won and by how much goals and stuff like that. But is this the complete picture? Will you be satisfied just by looking at this and saying that, okay, I watched the game? Most probably not. So computer simulations supplement this information with the complete video of the match, which you can for full 90-minute coverage, which you can watch and see that how the players move around, run around, kick the ball, score goals, and all the entire dynamics which is incorporated into the system. So you can consider that as the part of a simulation where you can understand the mechanism and interaction of molecules and look at them in real time. So together we get a complete picture of how the molecule behaves or how the match is played in particular example. To do this kind of approach, we need to resort to the laws of physics because atoms and electrons are too tiny and they live in the quantum world. So their interactions are dominated by the quantum mechanical nature. The problem with that is only for a very few handful of small systems we can solve the equations, quantum mechanical equations or the Schrodinger equations directly or analytically. For example, this hydrogen atom system where there is a proton, only there is a proton and an electron. But if we go to bigger systems, for example, multi-electron atoms or molecules, then this particular equation has no analytical solution. So we cannot solve it exactly in pen and paper. So the scientist Paul Dirac famously said that the fundamental laws necessary for the mathematical treatment of large part of physics and the whole of chemistry are thus completely known after the discovery of quantum mechanics. But the difficulty lies only in the fact that the application of these laws leads to equations that are too complex to be solved. And in most cases, as the analytical solution doesn't exist, human can't do it. We need computers. There are two ways to solve this problem. One way to use the quantum mechanics itself and make some approximations that can give some information about the electronic structure of the atoms and the molecules. But in case of the biomolecules, most of the time we are interested in the predicting the physical properties. For example, like their folding and unfolding or binding, unbinding, and these sort of processes, which does not involve chemical reaction or the transfer of electrons. In such cases, we can go to a classical model which doesn't involve the electrons but only look at the position of the atoms and their interactions. So we can make a model of which is known as a molecular mechanical model, where we treat the non-bonded interactions like the van der Waals and the charge-charge interactions, as well as the bonded interactions like bond vibrations, angle vibrations, and dihedral angle rotations, using some empirical expressions. And the parameters in these expressions are fitted or parameterized by uh, either quantum mechanical calculations on small molecules or maybe from experimental data. And combining these energy contributions from different interactions we can get the total internal energy of the molecule. And these internal energy can change depending on the configuration of the molecule. Now, once we can get the energy as a function of configurations, which is known as the potential energy surface, we can perform dynamics on that surface to understand their behavior. To give a little more overview of the potential energy surface idea, as this is a key concept in computational chemistry, you might have seen pictures of reactant, product, and transition state and a barrier crossing event, something like that, which leads to the going from the reactant to product stage. This is basically the same thing, but it is a multidimensional object because there is no one reaction coordinate, but it's all the conformational or the coordinate features of the molecule which we are taking into account in order to make sure that we are covering all the possible configurations that the molecule can exist in. And for different values, different configurations, we will have a different value of internal energy, leading to the this kind of plots which looks like a mountain, where there are high energy regions like mountain peaks, and there are low energy regions like the valleys. And you can consider the molecular configuration is like a rock in this mountain, which can roll down from the top of the peak to the valley based on the potential energy gradient. And that dynamics is actually determining the overall molecular motion of the system. These dynamics can be followed using time-dependent Schrodinger equation, which is known as the quantum dynamics, or maybe using Newton's equation of motion using the classical dynamics. This portion is known as the molecular dynamics simulation, which is which is one of the mostly used techniques to understand the behavior of biomolecules. There is also a way to sample this energy surface using Monte Carlo method, which is also used quite frequently. But to perform this calculation, it is often needed that we use supercomputers or computer clusters. Why do we need that? Because the integration of the equations of motion often requires the very short time steps to include the fastest motions that are present in the system. That includes the vibrational motions of the bonds between the heavy atoms and hydrogens. For that, we need a time step of 1 or 2 femtosecond. But the total computation, the total time of the event that we are trying to study may often go beyond microseconds or milliseconds, because that is the time scale where interesting biophysical processes take place, which involves millions and billions of time steps of integration, which is extremely difficult to do using just your laptop or a desktop. So here you can see one of the computer cluster present in the University of Illinois Urbana-Champaign, and you can see the stack of CPUs, which is much larger than size than a human being. And then you have all of these stacked together, and they are working together to increase the efficiency of the computational process. Here is another example of an Anton supercomputer, which is one of the fastest at present to do MD simulation related work. And here is the San Diego supercomputer, and this is the fastest supercomputer in the world, which is the Summit at Oak Ridge National Lab. These computers are almost like tens or hundreds of million times faster than the laptop or desktop which you are holding in your hand. So your laptop can give you some data, but it is very difficult to analyze a really interesting biological process in that single desktop computer setting. With that brief overview, I want to go ahead and describe some of the recent success stories of this kind of computational biophysics, and explain that how they helped in solving some problems, real life problems. So first, I will focus on the drug discovery against the HIV virus. So drug discovery for any disease is a long and expensive process, which begins with a target identification, which finds out the molecule at which the drug will bind. Then we go to the hit identification, and where we select the molecules which will be treated as drugs. And then we have to choose or select a few of those drug molecules that will eventually go to the preclinical trials on systems like mice or other animals. From after which we go to the clinical trials on the human, and after that the drugs are approved and they are available on the market. Each of these steps can take 1 to 3 years of time and billions of dollars, making the overall process extremely expensive and time consuming. Computational chemistry can reduce the time and cost by taking the first three steps into the virtual world. So here is an enlarged picture of those first three steps I described in the last slide. So at the beginning, we start with a large database of chemical structure or small molecule fragments which could possibly be used as a drug. And we also take another 3D structure database of biomolecules which can be used as the target, like the ones where the drug will bind. Taking these two things, a high throughput virtual screening is performed where, without doing any molecular dynamics simulations, just we consider a steric fitting of the drug molecule into the binding pocket of the protein to find out how efficiently the binding occurs, or get a rough idea about which molecules can bind. Once we can select a few molecules from that screening procedure, and this number can be in the order of hundreds, and then we take them to the heat optimization process, where we actually perform a molecular dynamics simulation, including all the dynamical motions and features of the protein and the solvent and the drug molecule, to understand and get a much more quantitative idea of the binding affinity or the kinetics of the binding and this kind of physical properties. From that, we can rank those drug molecules and choose the first few candidates for a in vitro tests and experimental trials and preclinical tests. And I will give an example of the HIV because this has been a particularly easy to understand example of this process. So HIV virus has this kind of a structure which has a lipid protein membrane in the middle of which they have the genetic material which is the RNA. And when the virus enters our cell, it puts the RNA inside the human cell, which goes through a lot of process into the integration into the host DNA, from which new viral particles are generated, and they become mature virus and come out of the cell. So the protease enzyme is used in this particular step where new infectious viral particles are generated from the small proteins. Here you can see the structure of the protease, and here it is complexed with an inhibitor, which is a drug that reduces the capability of the protease enzyme. When the protease inhibitors were introduced in around 1995, it has significantly reduced the mortality from the HIV diseases. So now you say that, okay, we have discovered an inhibitor. Now why bother about it anymore? We need to, because the HIV virus is highly mutable, so it changes it changes its sequence and the structure very frequently, because of which we need to always come up with new inhibitors at a frequent interval. So there are multiple efforts which has been taken in order to study this process in computationally, and I will describe one of those methods which is particularly interesting to me, and it involves a computational trick which is not possible to do experimentally. So here you can see the one of the ligand, which is say considered ligand A, in unbound state and in the floating in water, and here is the ligand in bound state. So this is the binding process which goes from this unbound to bound state, getting inside the protein and binding. So this is a very slow process, and if we want to simulate this in computer, even using computer clusters, it might take a very long amount of time and can often go from months of simulation time to get this thing done, which is not possible if you want to screen multiple molecules. So here they are using something called a relative binding free energy calculation. In this way, you suppose you need to know the binding free energy of one of the ligands, and you want to predict the binding free energy of another ligand which has a relatively similar structure. So what we are doing is we take the unbound ligand of which the binding free energy is known, and in the water, using a computational technique, we switch it to the new ligand by adding a functional group in the through the computational process. So this is completely impossible and unphysical to do in experiments because the molecule does not change itself without doing any reaction. But this is perfectly doable in computation because we can add new atoms during the simulation, and that's possible. We can do the same thing with the bound state as well. And then, as we know from the thermodynamic cycle, that the free energy is a state function. So if we complete the cycle, the total free energy change should be 0. And then we do some subtractions and things to use the value of this delta Ga and delta Gb, and we can compute the difference between this actual binding free energy of the two ligands. From there, we can predict the new binding free energy of the other ligands, unknown ligands. That is one of the approach which is taken to compute binding free energy. And here, to understand the mechanism, I will show you a video of a. So I'll start from the beginning so that you can see where the simulation has started. So here we start from an unbound state, and this is the crystal structure of the bound state, and and then when I play the video, you can see this during the simulation, the ligand or the drug comes in and then interacts with the protein, and the protein also changes its conformation a lot and interacts and goes through a lot of different intermediate states before finally reaching to the bound state structure. So these kind of dynamics is not visible in experimental studies, but using computation we can get this idea of the dynamics, and these are really helpful to design new drugs because we need to know that how that drug will interact with the protein. Yeah. So I will talk about another aspect, which is the protein folding, for understanding the physical basis of life. And sorry about that. Yeah. So the protein folding problem is generated from the Anfinson's experiment, where he showed that the native state or the folded state of the protein can go to the denatured or the unfolded state if you apply with some denaturant. But then when you remove that denaturing condition, you can get back the folded state and it can perform its function properly, which indicates that the uh, the information that is needed to fold the protein is already available in the sequence of the amino acids that are present in the protein. This leads to a dilemma which is called the Levinthal's paradox, which was proposed in late 1960s, which can be explained with this example. Say I have a protein with 100 amino acids, and there are approximately 5 states for each amino acid, which leads to 5 to the power 100 possible configurations to be explored before we can reach the final folded state. Because if all the possibilities are equally probable, then you have to go through all of them before we can find that which is the actual correct state. But if the biological systems tried doing that, then it would require a time which is longer than the age of the universe to fold a protein, which is not true because otherwise we would not be living our life like that. So how does it take place actually? So the most accepted idea currently is this energy landscape idea, which is similar to the energy landscape that you have seen before in this mountain-like picture where you have valleys and peaks, which is uh, mountains and valleys, and these lower energy regions which are in the valleys are basically correspond to different folded and partially folded states of the protein, and the unfolded states are high up in energy. So the the going from the unfolded to folded is basically going downhill through this energy landscape, so that these processes are kind of uh, like accelerated by the free energy gradient and not just a random searching over everything. Although one might understand that the native state or the folded state may not be the global minimum of the free energy surface, and there can be other low energy minimums which can lead to amorphous aggregates or amyloid fibrils, which is responsible for diseases like Alzheimer's and things like that. Here I will show you a video of a protein folding uh, simulation performed using molecular dynamics, where you can see this is the crystal structure of this small protein, and you can see this stretch structure, which is the unfolded state. And then when the simulation performs, the system explores various unfolded and partially folded states, intermediates, before finally going into the folded crystal structure, which it will reach very soon. So yes, now you can see it has converged into the final folded structure. So doing the MD simulation actually helps in a way that we can look at all the interactions that was happening during this folding process and can understand that which possible interactions have led to this particular folded state, and what is the mechanism of the folding, and what are the intermediates that are explored during this process. So with that, I will give a brief overview about our work which I did for my PhD research. And the first thing I will talk about is Hookstein base pairing in DNA and RNA. As you know, the in DNA, these two helices are intertwined with the Watson-Crick base pairing between AT and GC base pairs. But there exists some unconventional Hookstein base pairing, where this adenine base flips 180 degree on the along this glycosidic angle, and leads to this different form of base pairing structure. So this is very rare and can be less than 0.1% in the overall DNA population. But it is very important because it has been postulated that these Hookstein base pairs can play a big role in DNA replication and DNA recognition by small molecule drugs, or DNA proofreading by DNA damage repair enzymes like p53 protein, which prevents cancer. So it might be possible that these are the regions where these proteins or drugs bind. So it is very important to understand these base pairs and how these base pairs are generated, starting from a Watson-Crick structure. So we performed a molecular dynamics simulations for both DNA and RNA to see how the Hookstein base pairs form in both the cases. So here you can see that we have computed a free energy landscape, which is equivalent to this mountain valley idea of the energy landscape which I described before. So these kind of landscapes give a very clear idea about what are the intermediate states and what are the minimas and what are the uh, like the pathways which we can follow to go from one state to another. So as you can see, the Watson-Crick and Hookstein and a few other low energy states has been resolved in this particular process. And we can compare that with RNA and find out the differences in energy and the differences in the landscape topology, which gives idea about the mechanism of the transition in both cases. To solve this problem, we couldn't use just a regular molecular dynamics because the process was so slow that we estimated that it will take almost 60 years of simulation to actually look at one of the transition. So what we did is we added new biasing potential in the low energy regions of the valley, and we also applied a force against the potential energy gradient of the mountains, which is the high energy barriers, to push the system across the hills and to fill these valleys with, say, some fictitious water or sand to eventually flatten the energy landscape to increase the sampling. But then we need to use some mathematical techniques to rescale this energy back into to get the true free energy profile, which is like this kind of a free energy profile from this sampling. Okay, so that is one of the brief examples. Another one, which is a more recent work, where we tried to predict the mutations in the coronavirus spike protein using a molecular dynamics simulation. So we performed a relatively long simulation which was done over a few months, and then we analyzed the simulation using unsupervised machine learning techniques, which TICA and PCA, and molecular graph theory based networks. And with those approach, we could actually predict some of the residues which are present in the spike, mutations in which can lead to a modification in the dynamics of the infection and the binding of the protein, to the spike protein, to the human receptors. And on the residues that we found out, two of them led to the mutations, and lead to new strains of coronavirus. So the D614G mutation present somewhere in this region is has resulted in the Italian variant of the coronavirus which appeared in the March of last year. And this A570D mutation, which is famously present in the new UK variant, which is appearing recently, these two mutations were predicted from our model completely from a physical perspective and without using any mutation profile data or any population data, which was a great success of this kind of approach. So now I want to talk about some of the new frontiers or the new areas of the emerging areas of the computational chemistry. And I have must admit that there are like many different areas which I can talk about, which I will not have time to go over here. But I will only briefly mention a few areas which I am particularly interested in. So the first one I'm talking about is the machine learning based virtual screening of drug molecules for antibiotic discovery. So you know the antibiotics are particularly very important for treating mostly bacterial diseases and some other kinds of diseases as well. So the problem is we need to go over a large number of molecules to screen for the useful or maybe effective antibiotics. And it is hard to do computational calculation using a molecular mechanics or quantum mechanics for those many molecules because that's a very large like a molecular library. So here they used a machine learning based approach where they learn the effectivity uh, of those small molecules by training a neural network based on the structure of the molecules. So for example, this bond lengths, bond distances, and the connectivity of the molecule used as an feature, which is or a input set of data, which is trained into a neural network, and the neural network uh, predicts that which of them are going to be effective against a bacteria and which of them will not. And this training is performed on around 2000 molecules. Some of them has an can kill an E. coli bacteria and some of them cannot kill. And once this machine learning algorithm is trained, then it is applied to a large chemical library space of 100 million atoms, 100 million molecules. And this amount of large space is very difficult to explore using traditional computational methods. But this machine learning technique can quickly boil down this to a handful of molecules, which are called the leads, which go for the experimental verifications. So when this experimental study is performed, then it like they could actually discover quite a few, for example, in the range of 20 to 30 new antibiotics that are not only effective against E. coli but also against other other diseases like tuberculosis and typhoid and things like that. So this work has recently came up in the Cell journal in last year. So another endeavor is to going beyond the single molecule approach of molecular dynamics, because now we have much better computers than what we used to have 10 years ago. So now we can actually look at molecular interactions as we see in this video, where the receptor binding domain of the coronavirus spike protein is binding to the ACE2 receptor of the human cell, and multiple binding pathways of this process can be explored of two referent proteins. These were difficult to do earlier, but it is now possible, and it opens up a huge opportunity to look at protein-protein interactions. Here is another simulation where we are showing the coronavirus spike proteins, multiple of them present on the cell membrane. So this is the cell membrane region, and you can see the dynamics and the interaction between the different spikes. And so these kind of things are recently coming up where we can actually go beyond single molecule and look at molecular assemblies and even bigger like an organelle scale simulation, and we can understand the chemistry which is leading to these behavioral changes in those organelles. Sorry about that. Yeah. So another application is I will focus on the environmental side of designing a plastic degrading enzyme based on computational technique. So as you know, the plastic pollution is a menace of the 21st century, and one of the key components of the plastic that is undegradable is called polyethylene terephthalate or PET. Recently, there has been a discovery that this particular bacteria of Idonella cycliensis that can degrade plastic using a coupled enzyme machinery of PETase and MHase. So the PETase enzyme digests the polyethylene terephthalate into some intermediates called MHates, and these MHates are digested using the MHase enzyme to lead to small molecular parts. This is the mechanism of the process in which the MHase works by digesting the MHate into TPA and hydrogen peroxide. This mechanism was discovered using a computational simulation using a method called QMMM, where we combine quantum mechanics and molecular mechanics. So as you can see here, this is the M head structure, and these are the parts of the protein. And here you can see some proton transfer and bond forming and bond breaking is happening. And at the end, we release the hydrogen peroxide and the TPA molecule when the reaction is complete. So this is the catalytic domain of the enzyme, which is helping this reaction to happen. Now you might think that, okay, this is a video which is made by the artists, but that is not. So this video is completely the output of a computational program which perform quantum mechanical calculations. There is nothing there in this video which is like kind of made by humans. So this is only the coordinates of the atoms which are predicted from the quantum mechanical calculations that led to this video, and which led to the understanding of this mechanism of how this reaction happens. And this understanding will help to generate better enzymes or to design better catalytic domains which can enhance this process and can uh, like to move us one step forward in solving the plastic problem. So I want to give a brief idea about how it feels like to be a computational chemist. From outside, you might think that a computational chemist's life is beautiful because you don't have to go to the lab like an experimental chemist wearing like lab coats, goggles, and like closed-toe shoes and stuff like that. And you can work at any time, any hour, at any place, basically. But as in every endeavor of science, computational chemistry is also has its difficult periods and failures and things which doesn't work and frustrations. For example, your code might not be working, there may be a bug in your code, your theory might not be giving the correct result, and also your calculations may not be converging, and you need to redo them again and again to get a good idea of what this of your problem. Yeah. So to but to to go through this kind of processes, you need a good scientific background of both the quantum mechanics and statistical mechanics, which, as many of you are undergrads, and you will most probably learn in your future years if you take chemistry classes. So using this solid background of quantum mechanics and statistical mechanics will help you to solve your problems and can get you understand that where you are making your mistake, or why your research is not giving the optimum result you expect. So I hope the people among you who wants to become a computational chemist later on will be really enjoying this process and to solve new problems which are ahead of us in the future days. So with that, I want to thank my group at UC Irvine, my professor Yuan Andrichiwai and other group members. And this is the UC Irvine campus, and also the extended group in ISA Kolkata with Professor Ashmi Tiwari and Professor Amlan K. Roy. And I would thank you all for your attention, and I would thank the organizers again for giving me this opportunity to give this talk. And if you have more questions which you feel shy to ask here, you can obviously reach me in this email and Twitter. Thank you.
Thank you so much, Dhiman, for such an interesting talk. And we have a few questions
Yes.
which have popped up. So the first question is by Sudipta, and the question they're asking is, what is the basic difference between Monte Carlo sampling and MD simulation?
So I think I should quickly go back to that slide. Yes. So Monte Carlo sampling is a random number generation process In this way, you can sample different parts of the potential energy surface in a uh, like this sampling process will actually give you structures which are more probable due to their low free energy value or low potential energy value. And in this way, you can find, say, for example, these blue dots as you see here, these points can be found and depending on their probability. But the dynamics is actually missing in a Monte Carlo sampling. So it is not propagated along the time. So in molecular dynamics simulation, one performs the dynamics using the Newton's equation of motion. So you can follow the time steps. So you can start from a structure and move to the new structure as it would happen in a real life system in a cell or a chemical environment. But in Monte Carlo sampling, you only get a set of structures, which you can see that these are the possible structures that the system can explore, and these are the probabilities. So that is the basic difference. So the time component is missing in Monte Carlo sampling, but in Newton's in MD simulation, the time component is there, and you can follow the time sequence.
Thanks a lot for the answer, and I guess your doubt has been cleared, Sudipta. But if you have more questions, you can post in the chat box. The next question is by Abradev, and they are asking if the native state is not the global minimum, how do we explain that it is found more frequently in the native state than the global minimum?
Yes, that is a very interesting question and a very important question. So the native state is not the global minimum, but when the protein folding happens inside cells, it goes to this particular state, which is stable relatively for some amount of time. But if you are allowed to wait infinite time, then this state will go to this global minimum state, which is the amyloid fibril or something like that. But you must understand that the human lifespan or even the lifespan of a particular cell is not infinite amount. So if we can generate a machinery which the cell has already generated to get to the native state during the time, and say the protein will work for some amount of time, like for hours or a few days, and then it degrades or the cell dies, then it is perfectly possible to stay in that native state. Now, if you somehow cross these barriers by using some mutation or maybe some misfolding or some other problems, then you will eventually lead up to these structures. And it is almost impossible to come back from this structure to the actual native state, because these amyloid fibrils have very low free energy. So this is a design which is made by the cells to cope up with the system that we want to do, because we do not want a protein which will remain intact for like thousands of years, but something which will work during our lifespan to solve our problems inside our body. So that is why this thing is stays like that. But what your question is perfectly valid, that if we want to go to the lowest minimum energy structure, then this would be the structure that the system would go in. But due to the kinetic stability, it remains in this native state. And this native state is stabilized by some hydrophobic interactions like excluding waters and things like that, which I didn't go into much deep, which gives it some sort of stability for the purpose of its for the purpose it serves. And another quick point I want to mention is that for the functioning of the protein, you need to have some sort of dynamics. Like if you go to the lowest minimum energy configuration, then it might become a complete solid rock, and then it will not do the function that you are supposed to do. So that is also a biologically relevant reason to not go to the completely lowest global minimum. Yes, I think that's it for this question.
Yeah, thank you for the very insightful answer, and thank you for the question. We have another question by Shayantan, and they are asking how to calculate enthalpy of formation of polymers, taking into account the terminal and groups apart from oligomeric methods, which uses the extrapolation of results. I mean, is there any alternative way around?
Uh, I completely didn't follow it. Like oligomerization method of what? Polymers?
Yeah,
Like,
yeah.
can you
So they
repeat
are
the question
asking.
again?
I'll repeat. Yeah, I'll repeat the question.
Yeah.
How to calculate enthalpy of formation of polymers, taking into account the terminal and group groups apart from oligomeric methods, as in if there are any alternative ways around.
Yeah, I don't think I discussed it, and I am not sure about this particular area of science. I haven't worked on it, so I may not be able to give a good answer in this point. So I will not. I will skip this question, basically. So this is not my specialty.
Yeah, okay, Shantan. If you can reform your question, or if you have more questions, you can type it in the chat box or contact him by email. The next question is a follow up by Abradeep, who asked the previous question, and they are asking, as a follow up, how do we account for this finite time factor in the minimization computationally?
Um, Minimization. Did he ask the protein folding question, or like it is follow up?
Yeah, the protein folding
Okay.
question.
Yes. So the computational simulations are really like difficult to go to a scale where you can explore all the possible configurations in one simulation. So we are not there yet. So what we can do is we can start from a configuration and move to any either of them. So the video that you saw is basically going from one of these unfolded states to the native states. So this is not a minimization process. What we are doing is we are performing the simulations using Newton's equation of motion. So consider this is a rough landscape or a valley of like mountains, and you throw a ball into this. So what will happen due to the Newton's equation of motion and due to the physical laws, the ball will roll down this energy minimum and get into the low energy states, right? So that is exactly what we are doing in molecular dynamics simulation. We are not doing any direct minimization, but instead we are throwing the system in this energy landscape, and we are following that which pathway or which which are the basically which pathway does it take to go from an unfolded state to go to a folded state. And this is explored by this is like a a time series of the frames of the simulation. And this is not just like a kind of a minimization like which we just want to drag it the system down to a low energy state.
Okay, so Abradeep, I hope that answers your question. But if you have any more doubts, feel free to type in. The next question is again by Sudipta, and they are asking, is a chaperone protein mutation always required for forming the amorphous aggregates or amyloid aggregates or fibrils?
I would not say always, but because I am not very specialist in this area, so I cannot give a very detailed answer. But chaperones does help in protein folding, because in some cases the proteins are not doesn't have the capability of like folding the going to the native state just by itself, and it needs the help of the chaperones, and that's what the chaperones do, which is understandable. But a mutation in chaperone can actually lead to amorphous states and like aggregates, which is the most common situation. But in the time of stress in cells, these chaperone proteins can also like perform less well, like perform poorly compared to that it would do in the regular cell, particularly when after aging or when you become old. And in those cases, some of this can lead to misfolded structures and reduce their capability. That can lead to this formation of the misfolded structure. But you need to understand that it is not the exclusively the mistakes in chaperone that can lead to misfolding, because when you do some sort of a this kind of, if you see the Anfinsense experiment, so if you do this sort of a in vitro experiments, there you can see that this misfolded or the scrambled structures can also be generated by different kind of processes, just starting from the unfolded or misfolded or the native state, depending on the condition in which the protein is exposed to. So it depends on the, I would say it depends depending on the condition and what kind of experiment you are doing. That might lead to like either a mutation in chaperone or something else in changing in a condition to lead to these misfolded and aggregate states.
Thank you so much for the answer, and I hope that satisfies your curiosity, Sudipta. And if you have more questions, feel free to ask. And now I'll pass the mic to Shifalidi and Surajit if they have any questions to ask. So, yeah, Shifalidi or Surajit.
Thanks for the talk. Like it was a really beautiful talk. Okay, and I think
Thank you.
people won't have too much doubts considering how well you explained. But I would like to ask a few questions about
Yeah.
that. Can you uh, elaborate on how that molecular graph theory is used in? I think
Uh,
it was in
yes.
yeah in this process.
Yes. So in this model, what we do is we perform a molecular dynamics simulation, and then we check the correlation between the movement of different atoms in the structure. So we cannot choose all the atoms. So let's say we choose only the alpha carbon atoms of the amino acids, and then we compute the correlation between them. This correlation can be autocorrelation, which is the correlation of itself, and the cross correlation, which is correlation with the other amino acids. Once we have these correlations, then we construct a graph, which in this graph, so there are two parts of a graph. One is called the nodes, and our nodes are the location of the amino acids, and the connectivity of the graph, which are connections between the nodes, and these connections are the values of those correlations. Okay, so when we generate this graph, then we can compute different properties from the graph. For example, the property that we particularly focused on is called the betweenness centrality, which measures the importance of specific nodes in the graph by counting how many connections does it participate in joining different portions of the different nodes of the graph. So for example, if you choose this particular uh, amino acid, you might see that it is involving in multiple connections joining many different nodes, and this is the relative importance of each of the node. And then we try to understand that how this relative importance changes when this uh, when a conformational change happens in the spike protein, leading to the binding to the human cell. So from that point of view, we we can check that the difference of the importance of each of the nodes, and from there we comment on that. Suppose a particular node has lost or gained a value of importance, then we say that that particular residue was playing some important role, because like in the process it has become more or less important in the overall motion. So that is the basic approach that we took. You can actually look at our recent paper in sorry, this thing is coming up all the time. So our recent paper, which we've uploaded in bioarchive, you can get it from my Google Scholar or somewhere. So there you can get the details of the theoretical idea behind it. And if you have further questions after reading it, and you can of course ask me.
Yeah, thank you for the explanation. And I would like to ask one another question, general question.
Yes.
I think like there is a kind of maybe stereotype regarding theoretical chemistry that it is some kind of a supplementary field, and how is it improving? Like how is the image improving currently?
Yes. So it I don't know the condition in India recently, but because like when I was there, it was still like at least in ISARS, the people used to consider its importance. But there were, as I felt in India, there were few collaborations between theory and experiments. But now those things are coming up and become more and more apparent. The most important is that if you want to make a big discovery, you need to have a collaboration between theory and experiment, and that is much more prominent in US as I feel right now. And as I also worked in a few projects, and my friends have worked in a few projects of collaborating and getting good results. But now I believe that the theoretical chemistry or the computational biophysics itself is being considered as an important field because of the recognition that it is getting in different places. And uh, I would give for particularly one thing is promising is that I am seeing purely computational papers are getting accepted much more in journals like PNAS, Nature Communication, and like these big journals. And recently there has been a science paper just on computational modeling of a protein, which was kind of become rare in the 2000s, but now it's again spiking up because of the uh, when you get better computers, you can make a better predictive model, and when you can find a result which is kind of agrees with the experiment, then you can really bolster your claim that okay, what I did is significant. So in earlier days, when you have like a very few computer power and like simple, you have to make simple models and crude models where the predictivity was low. So now even we can come to the stage where the experimentalist can approach the computational chemist to ask for, like, see which problem I can work on, or maybe how to solve this particular problem. And then the computational scientist can bring up a solution or can propose a model which they can experimentalist can verify in their experiments. And in this way it works. So the collaboration is the most important thing, and that is what is mostly driving the new discoveries in chemistry at this point.
Thanks for throwing light upon this. I will pass the mic on to Sefer.
Hello. Yeah, thank you for this amazing talk.
Thank
For
you.
uh, this field, I do not have much experience with. This talk was like a very nice summary of uh, the general approach in this field and its possible applications. So,
Yes.
from what I have mostly heard, computational approaches, they generally require intense um, like structure, like you have to assemble a lot of computational power basically,
Mhm.
and hence it would be quite a bit expensive. So my question was, at what point do you decide that okay, the experimental approach would not be enough for us to get data, like in the drug development case that you explained earlier on.
Mhm. Okay.
At that point, why do we go for a computational approach, and would it be as expensive as the other alternative? I could not understand the alternative, or was you what was the approach earlier before advanced computational methods were discovered,
Mhm.
and what these what is the what are the advanced computational methods doing, and would the expenses not be too high? Like, if you are uh, if you are saying that the other approach, experimental, I guess, would have been
Mhm.
too much expensive.
Yes. So yeah, that's an important point. So what is the alternative is basically performing all these steps using experiments. So you choose multiple proteins and then throw in multiple molecules and check that which of them binds, which of them doesn't bind, and then you choose a target. Like, if you go back to the drug discovery process in say 1950s or 60s, in those time all the processes has to be performed experimentally. Someone will have to identify what is the cause of the disease, and then someone will have to uh, do the experiments to check the lots of different kinds of molecules and check their binding, and then administer them in a body and check their behavior and things like that. So the computer simulation part is helping in a few ways. One of them, which is we can get a molecular level picture and the understanding of how the binding happens. So once you look at something, you can get a much better idea about the what is the next thing to design. Suppose you make a small molecule and you can see that it is not binding properly, but you can also see that what are the interactions that are missing and which you can include in that molecule that can eventually lead to a better binding affinity or a better binding drug. So that is one thing which accelerates the process. Another approach, another advantage is that if you replace the experimental part with a computer simulation, then you will get a lot of saving in terms of time and as well as the cost. A saving of time is actually equivalent to saving a cost because you don't have to pay a lot of people to do a lot of work in the industry and stuff like that. And also, once you buy a good computer, or say you invest on a big supercomputer, it is a one time expense that I can understand. But later on, that supercomputer will be usable for like at least 10 or 20 years, and during that same time you can use it as much as possible to compute many different properties and like many different screening procedures. Because it doesn't require any kind of a, what do you say, like disposable chemicals. Basically you don't have to make like buy resources every now and then and employ people to do this kind of work. So it saves money in terms of both the salary of human and also in terms of buying expensive uh, experimental instruments and chemicals. The third thing, which comes from the chemicals, is that if you perform experiments on 10,000 or 20,000 molecules or maybe a million molecule, you can possibly, if you have like an infinite amount of money, you can do that, but it will lead to a huge amount of environmental pollution because you need to discard those chemicals which are generated as a byproduct of those experiments. And these three problems are basically taken care of the computer simulation. Although computer simulations require power and you need to give energy and money, which is of course it's needed, but it is much less expensive when you are doing it at a very large scale. Like you are taking in thousands and millions of molecules, and you are doing computation on them. Then it is much more like I would say it takes much less expense compared to an experiment, and it also helps in preserving our environment for avoiding this kind of polluting reagents to coming out in this environment. I hope that answers it, but please ask if that is not clear.
No, this was a great explanation. So I understand that the general approach is that had I been doing it experimentally, unless it's a small scale project, it would involve a lot of reagents which would be harmful to environment and an immense amount of manpower. So
Mhm.
instead, when the computational power balances it out, like it becomes much cheaper, and that happens on a large scale project, then
Yeah,
the
and a
science
large scale
has
project is
actually.
necessary because for example, you if you just start with a small set of molecules, you might actually miss the actual most best quality drug, right? So, if you want to go over a larger space of molecules, then you are more likely to find a better drug, right? So that's one of the aspects which you need this.
So the main merit of this computational approach is that at every step you get to see what your existing properties are in a molecule and what its merits and demerits are, and that helps you decide in what direction you should go next instead of just randomly making molecules and stuff.
Yes, yes. Because in experiment you just see the structure and just the binding affinity or something. Like it's just a kind of a black box sort of thing. And you can actually look at the molecules if you do simulations.
Okay, thank you very much.
Thank you for the answers, and thank you, Surajit and Shefali, for the questions. I had a small doubt, which was about the Hoxton base pairing. So I was wondering how different is it from the Watson-Crick base pairing, and if it does happen, is it like uh, can the enzymes or the proteins which act on DNA bind normally, or like what is the difference between these two? As in the binding of proteins are different.
Okay.
Uh,
Okay. Yeah. So this is the problem which we are looking at right now. So in our research, which we have done before, we compared just the free energy difference of the Watson-Crick and the Hoxton structure and how probable each of them, and the kinetics and this kind of things. But there has been a recent study which shows there's the P53 protein, which those who study in biology might have heard about because this is preventing cancer. So if there is a damage in the DNA, then the P53 protein comes in and corrects those damage mutations and make it like a proofreading kind of process. So if this P53 protein binds 6 times more strongly with a Hoxton base pair compared to a Watson-Crick base pair. Now, this has been observed experimentally, but now the point which we want to understand is that whether a Hoxton base pair is formed automatically in a region of DNA where there is a mutation, and whether this guides the P53 protein to specifically bind to this particular region. So this is like a big scientific question which is not known yet, and we are hoping to explore this in, like, at least I will might try to do it in my future years of my PhD, to study this kind of this particular question, like, is the Hoxton base pair playing any role in the binding, uh, and in the recognition? Because this is a postulate, because people have found crystal structure of these proteins bound to DNA, and they saw the Hoxton base pairs are there in the crystal structures. Now, whether that Hoxton base pairing is playing a dynamical role in the binding, that is a question which is not fully known, and that is something which we should explore in future.
Thank you very much for talking about such an interesting and also for working on it. It's really such an exciting field. And as we don't have any more questions, as an ending comment or an ending question, I'd like to ask you, or request you to give some advice for undergraduate students who are thinking of making a career in academia. What advice would you like to give to them?
Like I would say just the basic things basically. So you have to perform well in your like an academics in your undergrad, and then go to do a PhD from a good university with a good PI, and then like a postdoc, and then you can, if you want to become a professor, you can move to the do a postdoc and move to faculty job market, or if you want to go to industry, which I was not very much aware of at the point when I started my uh, journey. So now here in US, I came to know more about the industry opportunities, because a lot of very important research in the field of drug development and also biomolecules are performed in the industry and in companies where they develop these new therapeutic methods, and computational chemistry also plays a big role in that place. So the next big market of the computational chemists are going to be in the industry, as I can understand. And there are different technologies that are also being used in industry. And if you want to become a professor and you go into academia, then there are a lot of open fields of research, which I mentioned only a few of them. And there are now this quantum computers are coming up, and we do not know that how much like new frontiers it will open up, and what kind of questions we might come up with. So one thing you should always understand is that you should do something which you enjoy. Like this particular problem, like when I first saw the move, like a video of a protein jiggling and wiggling in like water, and that was like the a big step for me. Like I realized that okay, this is what I want to do. So I can see the mechanism and I can see the reactions happening in a video. But it actually not only the video part. There is a much more quantitative understanding which you can get from the theoretical background of the this method. And um I would particularly say that be open-minded and to new opportunities. So when I joined ISAR, I had no idea about computational chemistry, and I was like determined that I will never do anything to do with computers, because I hated computers. And but then during my computer science courses in my first year, and also by working in internships and other projects, I gradually become interested in this area, and now I think that I'm not going to leave this field anymore. I really like this one. So depending on like your interests, and your opinions can change, and that is good. Like uh, that there is no problem with that. And you have to find your interest, and you can change your field even later. So you should be open minded. So you should not be thinking that okay, I joined ISAR because I thought that I will solve some maybe the mystery of black holes and something, and I will never do anything other than that. That's not true. If your passion is there, you can go for that, but you should always be open minded and look for new opportunities and new different areas which are coming up. And I personally think that there are like uh, as the new advent of new computer technologies, I think new problems will come up, and very soon maybe you can go from this organelle scale simulations to a more of a cell scale simulation, or maybe an actual organ scale simulation. And I don't know whether this is actually possible, but these are like many long future goals which one can think about. So there is endless opportunities. And like if you want to ask for anything specific, then I can give you a very more specific idea. But overall, like you have to go through all these processes of science and like application and doing PhD, postdoc, and stuff, and then you choose your career in science as a scientist if you want to be a scientist.
Thank you very much for the insightful advice and talking about this really exciting field of uh, uh, molecular docking and chemical simulations and computational chemistry in general. So, so viewers, I would like to thank you for attending this talk, and I'd also like to thank Dhiman for coming and giving his time and waking up early in the morning and helping us out in this event. So we'll see you in the next event, everyone. And so I'll stop this live stream now, and I hope that you have a good evening.